Hello, I'm Robert, and this is again about my paper. I'll just put it up here about why NASA might be legally required to sterilize Mars samples to protect Earth's biosphere. So now, please see my previous video, which I'll put a link into the description if you haven't seen it, and you can have a read of this, which is in the video, will be in the video description. So now, in the previous one, I got as far as explaining why they're likely to have to sterilize it. By sterilizing it is an option. And the reason being, just very, very quick summary, and the reason is because we don't have the technology to contain something like mirror life returned from Mars. We simply don't have that technology yet. And once you've gone through the legal process, NASA will not build a sample receiving facility. They won't start the build until they know that they actually can build it with present day technology, and they won't release the funding. And the other thing is that it's not actually guaranteed in advance. It's not like the Europeans, like the sample return studies, where the remit is that you have to return a sample safely. It's not guaranteed that the end point of the legal process will be that it is permitted to return the sample. This depends very much on how they interpret the precautionary principle. If they take Carl Sagan's point of view, that it is not permitted to take even a small appreciable risk with 1 billion lives, then we don't currently have the technology to return a sample from Mars. So they would have to wait to the end of the legal process to see if they're actually permitted to build the sample receiving facility, and having done the sample receiving, then it takes 11 years to build the facility, and that takes us to 2040s. And the various studies for possible sample receiving biosafety cabinets and biosecurity laboratories and so on, they don't meet these standards, which are that at a minimum requirement, it's not permitted to release a single particle of larger than 0.05 microns under any circumstances. That is likely to be a legal requirement, because we have bacteria that can go through 0.1 micron filters. So it's not likely that they say that it is permitted to use filters that will let Martian microbes get into the terrestrial biosphere. So for that reason, it's likely to be a minimum requirement to at least that. And then I talked about ways that maybe it's a possibility that other forms of extraterrestrial biology could go well beyond that, to even the requirement might even, on review, based on research for the last 8 years, be 0.01 microns, which is even more of a challenge. And we just don't have a way. If there's something like mirror life on Mars, mirror photosynthesis, and I talked about how it would not be possible, the life living in a salty brine on the surface of Mars could not be transferred back to Earth via meteorites. It's the wrong place to do that. And also talked about how Mars could have a self-perpetuating swan song Gaia. And if that is possible, where the photosynthesis which on Earth keeps Earth from getting too cool, on Mars it could keep it just above habitability as cool, so it is just it could take what could otherwise have been a habitable Mars with quite a thick atmosphere, and it could be the reason why Mars has been so thin for billions of years, at just close to the triple point of water. It could be that without life, the Martian life atmosphere would be much more habitable. The Earth's atmosphere is not optimal, it's suboptimal for life, because a warmer Earth would actually have more life on it. And you can argue that. It's not clear that it's optimal, depending how you think about it, but there's at least some 10 degrees, 20 degrees warmer than this could well be more optimal for life. Gaia does not necessarily keep the planet totally optimal. It is close to optimal, but then with Gaia, with Mars, it could be well suboptimal for life, because the photosynthesis would take it out, and I explained all that in the previous video. So that's where we got to so far. So in that case, given that background, it seems quite possible that Mars could have life there. So the simplest approach is for NASA to just sterilize the sample return. And I talked about that now using miniature X-rays, nanoscale X-ray emitters, is a very low power way of sterilizing a sample during return mission from Mars to Earth. So by the time it gets back here, it's sterilized. You could just do aero capture, and it would be a very easy way to return a sterilized sample, and would save hundreds of millions of dollars to do it that way. And I think that's what NASA will eventually do. But they probably have to go through lots of all this process. So I'm just anticipating what they're likely to decide. And I'm saying that it's good to decide this now, right away, because it saves an awful lot of work if we decide in advance that we're going to sterilize it. However, it would just save years of work, and it would also mean that we sent something much more appropriate in the form of the satellite we send there and sample return container. However, I also argued that there's a possibility to turn this around into a mission that could be very interesting for astrobiology if you accept the argument there that there is likely to be life in the brines found by Curiosity. So, sorry, this one. So, these brines, I mentioned up there that these brines in the surface of Mars, where are they? These ones. Then there could be life there, possibly in biofilms, Martian life in Martian biofilms, could retain the water into the daytime, possibly using cheotropic agents in order to speed up the metabolic processes. And then it's just possible, I'm not saying this has to be the signature of life, but the oxygen evolution that Curiosity discovered of more oxygen in summer, less in winter, and less oxygen when the dust storms. This is not an impossible signature for life, which also some of the researchers said about it themselves. So, like Trainer was the lead author for the Curiosity oxygen results. People in the community like to say that it would be the explanation of last resort, because that would be so monumental. The abiotic mechanism is plenty, and to rule out first before leaping to any more sensational claims. So they're not saying it's life, but it's not impossible it is. And my Gaia reasoning, Swan Gaia reasoning, is extra reasoning to support the idea, together with the offset chiral circadian rhythms in the Viking labelled release, offset by 2 hours, which suggests that Viking did discover life. So if we think that there's a high probability of life on Mars, both in these surface habitats and in the dust, then there's another way we can optimize the mission to return that life. It's not too late, even though it's not designed to find past life, present day life, then we can optimize the mission first by the caching strategy. So I suggest that we can leave the dust colours on the surface of the rovers, and so there's definitely a whole lot of dust that keeps falling down all the time, and especially around magnets. If we were designing these samples from the sample containing tubes from scratch, we might put a magnet in the bottom, but it's too late for that. But they could put a sample tube on top of magnets, sitting on top of magnets on the rover. There may well be magnets that they could put on top, and then the dust would accumulate into tubes preferentially. And then the other way they could collect the dust is by just leaving these tubes vertically in. So there's the suggestion to leave it there in vertical. So I suggest we leave them in vertical position to gather dust, unstop, uncapped, and then cap them when the rover returns. And so that would give several months just waiting for the dust. And again, that one, and by having, and you could have other ones along the way. And these could give you an idea of get samples of dust from different times of the year, different years. And the reason is because Martian microbes, if they exist, may well have developed to propagate in the dust. And they may find little niches in the dust. And I talk a little bit about propagation strategies they could have developed in my paper that could lead to them being in the dust. So that's one thing that you can look at. And then it's also very good to have samples of the dust, whether there's life in it or not. It will help us to understand forward protection as well as to what kind of material the dust is. And it may vary during the year, and different times of the year, different in the season, what the dust is. So having these sample tubes where you could actually measure you'd have different layers of dust accumulating it, then you could either put the dust in the bottom, and then later on the sample goes back and picks up the tube and then adds a sample on top of it, or you could put the sample on top of it and leave it open and the dust lands on top of the sample. Either way, I think it would be of great interest to to let dust accumulate inside the tubes. It wouldn't be that many, but just even a few dust grains would be of great interest from different times of the season from Mars. We don't have any dust from Mars. As I said before, this is a habitat that has not been transferred to Earth. However much people talk about Canadian geese flying from Canada to the United States, it's not the same. We have no dust and no brines, no salt from the surface of Mars. Whatever life there is in there has not been transferred to Earth. Probably never. So it's very great interest to collect the dust and see if there's any life in it, and if there's not, then still discover whether the dust could transfer life. There are the chlorides mixed in with the dust, but they only become activated by UV, and without the UV in a thick dust storm, then life may very well be able to propagate around on the surface of Mars. And it's not impossible that Martian life so sporulates and produces these capsules which would be covered maybe in something. It might even use iron oxides. So I suggest that in my paper. They might gather the iron oxide like some microbes do to protect themselves and make a little shell of iron oxide particles, tiny little dust particles, and then that could be blown in the wind. That's another possibility. So in effect, some of the dust particles mightn't be dust particles. They might be small microbial spores that have gathered a shell of dust around them, and there are some microbes that do that on Earth. In the sea, they don't do it. They don't do it on the. I couldn't find any examples of airborne microbes that do that, but certainly sea microbes in the sea and in water, and they do that. And so microbes in these brine layers could do that, and then they could expose themselves into the dust. And then the ones in the sea do get transferred in the atmosphere on Earth. So this would be very analogous to the way seaborne life on Earth can be transferred to another aquatic habitat, as happens, for instance, in Antarctica, that some life can be transferred from one pool to another. So this could happen on Mars. So if that's happening, we might find them in the dust. And so that's one possibility. And then so that I think would greatly enhance. And this doesn't need any change. It just needs changing the caching strategy, not any hardware. Now, another thing that would be of great interest to increase the interest for finding present-day life would be to add a scoop to the retrieval rover, ESA's Fetch rover. So ESA's Fetch rover, that is just from a 3D animation. It's just meant to be a couple of things that you pick up, and that's all it does. Its only job is to find the samples, pick them up, and put them into the sample cache receiver, the sample caching container. I suggest that a modification of this to add a scoop. So maybe one of those two things has a little scoop on the end of it, or just attached to it, in such a way as to not interfere with its ability to pick it up, but it would also permit it to somehow scoop up a little bit of dust. It's a very similar pattern to the Venus, to the Viking scoop. If we do that, then my suggestion is that we take the sample container, orbiting sample container, and we add an extra plate on top of it, and then we scoop a sample of dust on top of it. Now if we dig deep enough, then we can scoop up, if we can do it, if we're just lucky enough that the sample Mars Ascent vehicle is in the sand dunes, in an area that has these damp patches, these salty damp patches at night, then we could actually sample those and put those on top of the container. And this would be of great interest for astrobiology, either way, whether there's life there or not, because we don't fully understand the Viking labelled release. So we would take a little bit of the dust, identical to what Viking sampled, and then bring that back to Earth. And bring it back to, so then the question is, where can you bring it back to? So we can't bring it back to the Earth's surface. As I said, we just are not going to complete the legal process in time, whatever you think about the precautionary principle. So the question is how to examine it, but I'll talk about that in a minute. But that is my proposal, that this is modified, and this is very early. So, for instance, senior moment. Not really. I've always been absent-minded like that. Chris. Chris. What's? Sorry to Chris. Chris. Chris McKay. So Chris McKay proposed a sample that the best way to do a sample return from Mars would just be scoop up a patch of soil and a grab of dust, just one day on the surface tops, and return to Earth. In terms of astrobiology, that's as interesting as anything. And it's really just a technological demonstration to show that you can return something from Mars to Earth. There's a possibility you find something interesting. So I'm suggesting that we basically do that. We combine that with Perseverance. We have all the rock samples of great interest to geologists, and then we have the sample of dust which the astrobiologists would possibly be salivating over. And that might well be more interesting to them than the samples. Although the samples would be of some interest, they'd be of comparable interest to the Martian meteorites, because they're very unlikely to actually sample actual recognisable life from past life on Mars. They're likely to be ambiguous, as I explained, because of all the various processes on Mars that are sure to have deteriorated, and because of it's confused with the organics from comets and meteorites, which are far more prevalent on Mars than the past life organics if it's there. So that would greatly add to the interest for astrobiology, in my view. Again, astrobiologists, please, what do you think? So that's the would that greatly add to the interest of the mission? I think it would. So my suggestion then is, but what do we do if we've now got a mission which now think is of great interest for astrobiology, and especially, and then I just want to mention one other thing. Oh yes, one other thing I'd like to mention is the possibility of getting it more interesting for past life. So now if we look at crater here. Let's find the craters. Oh, it's going to take a while to find it, sorry. I'll also mention the craters, no surprise. How am I going to find this section? Recently excavated craters. There we go. Because I forgot to put that into my extracts. So, now this is for past life. Then there are many recently excavated craters on Mars. And some of them, they've actually discovered the formation of craters on Mars. And some have been formed since we started observing Mars from orbit. Now these would be of great interest for past life on Mars, because they excavate the surface to quite a great depth. So if some of these newly excavated craters happen to be in an astrobiologically interesting region like a delta, then that would be a very good place to send a rover, because you could go around the edge of the crater looking at whatever depth interests you. And so this shows how we can actually identify bits. The mission to Mars, Perseverance's target has not been selected on the basis of finding craters like this, but it's a very cratered region, just lots and lots of small craters, and I work out that there would be, of the order, that the youngest, that is based on this analogy, that there's likely to be 200 small new craters within 5 kilometers of the sample depot site, in the range from 16 to 32 meters. And the youngest of these, within 5 kilometers, is likely to be as young as 50,000 years old. That's very young. That is pretty much pristine. It wouldn't have been deteriorating much in the cosmic radiation or the solar storms. So, and at the rate, and that would excavate the surface to a depth of several meters. A depth between 2 and 9 meters. 9 meters is very deep. Based on 7 newly formed craters, they were between 2 and 9 meters in depth. So it's not maybe 4 or 5 meters deep crater. There could easily be a 4 or 5 meters deep crater, only 50,000 years old, within 5 kilometers of Perseverance when it lands. So main question is, how do you find it? How do you find such a young one? Dating from orbit would be quite hard. So a crater with a sharp rim like that, this is the process of degradation. Very sharp rim like that would suggest it's quite young. And then I suggest the Mars copter, it can fly to a height of 5 meters and a distance of 300 meters in a single flight. It takes 3D high-resolution images in between orbital images and images taken by the rovers at ground level. And this would help to find the crater with the sharpest features within 5 kilometers, especially if the rover could go up to a local elevation, so that it's just 5 meters above, so as to go above the nearby rocks. And it's on an elevation you could see maybe quite a way. Just fly 300 meters is not that far. It's the nearest one maybe 5 kilometers away, but it would be able to spot, and it would be able to spot newly excavated craters. And it could maybe determine which is the very youngest, even down to sort of the 50,000. You might even be able to discover the one that is only 50,000 years old. And then we can target that. And the youngest crater in the whole of Jezero crater is likely to be around 2,000 years old. But 2,000 years old is too young, so there was a possibility could Mars Reconnaissance Orbiter find a newly formed crater using before and after images, and find one that has actually formed within the time of observing it. That's another way to find a newly found crater. But the youngest one is likely to be 2,000 years old by my calculations. Please check and double check if you like. And so if that's right, then it's not going to be that easy to find one that is actually, we know for certain is new because we've seen it form. But we are very likely to find, there's likely to be one that's 2,000 years old that is over the larger region, and if we could find that somehow, then we could just micro-adjust where it lands, maybe. But they've already decided to target ellipse. I don't think that's going to change. But depending where it lands, it could possibly be one as young as 2,000 years near it. It's likely to be 50,000 years. So that's the suggestion, that we could have a more interesting past sample as well. Whether that could be interesting enough to be actually degraded by sterilization from Mars to Earth, and maybe it could be young enough to be degraded significantly. So then, if it's only 2,000 years old or 50,000 years old, that's not that much ionizing radiation. It's not nearly enough to sterilize a terrestrial microbe. So the amount of radiation that you use, even with X-rays, to sterilize back, I've got to say that the advantage of X-rays for sterilization, I said last time, is it's tunable. So you could tune it optimally, what frequency of the X-rays, in order to have the minimal effect on the geology and to sterilize the biology carefully in just the way you want it to be sterilized. So there's obviously room for some experimentation with the X-rays, find out what's best for geology and for astrobiology. So, based on all that, we may well be able to return something much more interesting than the sterilized sample that I talked about last time. In that case, it'd be worth the extra few $100 million to return it to above GEO. This is my favourite location to return it to. So that is, so I'm going to talk in a little bit of detail about that, and then I'll finish this, and then this will be enough for now. Yes, I'll do another video later about the, I think I've said enough. I might do another video. I wanted at some point to talk about the astrobiology and the hazards for Earth. I've got looking a lot of detail into that. I would like astrobiologists to have a look at that and double check what I've done there as well. I'll probably do that as a third video at some point. But you'll see all this in the paper. So now if we look at the this is the modification of returning to above GEO. It just stays there. It doesn't come down to Earth. So that's meant to be the satellite above GEO. It doesn't do the last little bit. And then we teleoperated it in situ. And this is very fast, very low latency, just to above GEO. And basically, the idea here is we return the sample to above GEO, and then we do basically a Mars mission to above GEO. We have the equivalent of our Mars rovers, but we'd just send instruments to this satellite above GEO with very low latency. And so we can send a new instrument. If we find something interesting up there, and we need to send such an instrument, we can send it immediately within a few weeks or whatever, depending how easy it is by then, by 2030s, to send material to above GEO. It's probably very easy. and we could return it to a big satellite. By then, we could maybe even return to one of Elon Musk's huge 100 colonist size spacecraft by then. And because he claims that it's going to be very low cost. And so you could just buy a second-hand one, as it were, and send it up there and use it as you're receiving the laboratory, a huge one, that you'd examine via telerobotics. Anyway, whatever method you use, this would be a very low-cost way of doing it, because what you do, the advantage of doing it this way is you do everything as you only pay for things when you need them. So you return the sample to above GEO, you send a small satellite up there, or whatever you've got, you then sterilize half of each sample, or a quarter, or whatever's a reasonable amount, and you send that back to Earth. And then people look at, the geologists look at that, the astrobiologists look at that, because you'll be able to tell a lot about the biology if it is there. Then depending on what happens, you might decide just to sterilize everything, and then you just convert to the sterilized mission with just one extra flight that has been done in order to capture it. And then you just say, oh well, we're going to sterilize everything and return it, and then that's it over. But if you decide there seems to be life there, if you look, for instance, a sterilized subsection of that dust sample, and you see what seem to look like, you actually examine the morphology of what look like living cells, and you detect the organics of living cells, and you detect what seems like astrobiology, then you would then study it in orbit. You send instruments up to that in orbit. If you discover that there is mirror life in that, then you would not return it to Earth. You would take extreme caution, and then you take all the precautions needed depending on what you've discovered there. So you adjust your precautions according to what is there. And this saves $500 million on the receiving facility, because you don't need to build it. And then it saves the entire legal process. You don't have the precautionary principle problems, because by the time, if you do discover life there, you know what it is. And then you would have a much more streamlined legal process, because you could say, we have discovered X, Y, Z, and this is safe to return, so go through a much faster legal process. And then if you've got mirror life, then you probably don't return it at all, because I think just about everyone would agree that we shouldn't return mirror life to Earth, at least until we've got something much safer than what we've got so far. So we do it based on what we find there. But we've got to be careful, because you might find, for instance, some very feeble form of RNA wildlife, and it's not going to be able to compete with that life. It's not mirror life, but there might be, along with that, mirror life, for instance. And even very feeble mirror RNA wildlife could be quite significant effect on the Earth's biosphere. So you have to be sure that you've looked very carefully at everything there, and then once done that determination, then you can return it to Earth. So that is what I think we should do. And so I then went into some detail as to where we return it. And the best place is the Laplace plane. So the Laplace plane, this shows the inclination, and this shows the semi-major axis. As you can see, it goes up to higher and higher inclination, up to about 23 degrees, or some 22 degrees I think it is. I can't remember what it is now. So this is the same plane as is used for Saturn's rings. Saturn's rings are not in the equatorial plane of Saturn, they are in the Laplace plane. And the Laplace plane is due to the interactions of the gravity of the planet and the gravity of the Sun. And then this looks at the possibility of high area to mass ratio particles, which are also affected by the sunlight and pushed around by sunlight. So now, if we return a sample to the Laplace plane, so the geostationary orbit is in 0 degrees inclination. This is the angle of ascending mode, and the Laplace plane would be in the 0 angle of ascending mode, and it would be in about 7 degrees. No, what is it? 7 degrees, I think, of the inclination. So now, if you look at the evolution, if you had an adverse accident, you've got to be careful about what happens if, for instance, you send a supply mission up to up to send some instruments to observe it, or to retrieve a sterilized sample to return to Earth, and something goes wrong and it crashes into your sample-contained satellite. So it's got to be somewhere that's safe for that. Now, it can't be on the Moon, because the Moon has to be kept safe from extraterrestrial life, according to the Planet Protection Mission so far. In any case, the Moon is so far away, it's much easier to study it above GEO. It can't be, obviously, in LEO, because if something crashed into it, then it would just go off, contaminate other satellites, and anyway, its orbit would decay. A middle orbit you would think might be possible, but above GEO, in the Laplace plane, is by far the best. Now, if it's in the Laplace plane, then it can't get back to zero inclination. If it's other planes, then you find that it does get back to zero inclination eventually, even if it's quite high above GEO. But in the Laplace plane, so this shows these are particles with very high area to mass, very low area to mass ratio. These are very high area to mass ratio. For a very high area to mass ratio, it could go all the way up to 30 degrees, 40 degrees, even nearly 40 degrees inclination. But it stays within, and then it shifts back and forth in the ascending node. But it never goes down in this way to zero inclination. So that's the advantage of the Laplace plane. So I recommend that we return it to a satellite in the Laplace plane, so that anything that is shed by it, however, even if it's light insulation material or something like that, which also might just flake off because of sunlight, then it doesn't matter, because it will never come anywhere near equatorial orbit. Then if you look at the evolution of eccentricity, it only evolves to 0.4, short 0.0545. And so again, it's not going to hit Earth's atmosphere. And that just goes back and forth, and 0 to 0, but that's very high area to mass ratios. So this seems a very safe place to return it to. Now, we can return it from Mars. This is one that's actually a suggestion for a way to return it at very low delta V. So instead of returning to Earth, you can do a flyby of Earth, then a flyby of the Moon, and then you end up in a lunar direct retrograde orbit. This is an orbit that has the same period as the Moon, sometimes goes beyond the Moon, sometimes goes internal of the Moon, inside the Moon relative to Earth, and it actually orbits the Moon in the reverse order of what you'd expect. And so that's so it's called a distant retrograde orbit, a lunar distant retrograde orbit. So we could capture it into lunar distant retrograde orbit, and from there the delta V to get back to GEO is much less than if you try to come directly from Mars to GEO, which would require a very, very great deal of delta V reduction. To an Earth flyby, a lunar flyby, end up in DRO. Now, there's an even neater way of doing it, and this is use a halo orbit. And now this is going the other way. It starts from the Earth-Moon L2 position, and then you've got this thing that sort of spirals around the halo orbit. It's a halo about the L2, and if you start just beyond it and leave something there, and over time it will gradually spiral out further and further and further and further away against quite a lot of delta V, as it eventually escapes from the Earth's gravitational pull. So the idea is to use one of those. So you start off in LL2. So this is going the other way, from Earth to Mars. Go past start from LL2. That's the stationary orbit, balance of the Earth-Moon's gravity beyond the Moon, the far side of the Moon. That goes on, and then it goes out this halo orbit. And then to get to Mars, it would then do a flyby of Earth, and then it heads off to Mars. That's going the other way around. That would be a good way to come back with very low risk. And then another way is this is even much lower risk. You have the Sun-Earth halo, which then transfers to the Earth-Moon halo, which then transfers to LL2. Once you've got to the LL2, the far side of the Moon, then you can get to LL1 on the near side, and then you can go into you can go into orbits from there that get it with quite low delta V. You have a necessary, unavoidable delta V about 1 kilometers per second to get to GEO, but you can really reduce it quite significantly, the amount of delta V, and you can do that over a series of time, because they're going to use electropropulsion with the ESA satellite. So it'll have plenty, given enough time, it can do this without needing to add any more fuel to it. So I suggest that the ESA satellite, the ESA fetch orbiter, we do this. We use it to return, probably this is the best of all, but the timing, you have more opportunities of this, and then you have even more opportunities probably of this. But this is the best of all. And it could target one of these orbits and end up in that, and then in that. That would be very low delta V, and then get it back to, and it would easily have enough delta V then to get back to GEO instead of trying to go back to Earth flyby. So we need the space engineers to look into this. Is this correct? You haven't missed anything out. As far as I can see, this is a safe way and an easy way to return a sample to above GEO, and then we'd send a satellite from Earth up there to meet it. So that is the main thing I wanted to say in this part of the talk, and so that's it. Just the idea. And so then the idea is that you study it by telerobotics there, and then you decide whether you've got to sterilize part of it. You sterilize part of it, and within a few weeks you will know. You may make a decision. Oh, we're going to just sterilize the whole lot, and that's the end of it. But then you look and you discover that actually the interesting organics, what looks like life in the dust, for instance, or whatever it is, something interesting, you decide we don't want to sterilize this. Then in that case, then the satellite you send up there becomes the nucleus of a much larger facility. But you build whatever you want as you need it. You always send from Earth to Mars, to Earth to the satellite. You never send anything from the satellite to Earth without sterilizing it first. And this completely protects Earth biosphere. This entire process could be done within the Outer Space Treaty without invoking any terrestrial environmental laws. And I think that Mars colonists should want to know this as well, because if there is mirror life on Mars, then it's very difficult to see humans colonizing Mars, because the spacecraft going to be going back and forth between Earth and Mars, and how can they manage, with this huge big 100 colonist spacecraft, can they stop, can they make it impossible to return mirror life from Mars to Earth? There's no way they can do that. So we need to know, is there something like mirror life on Mars that simply shouldn't be returned to Earth under any circumstances? Because this changes our future plans. Now, if you are keen on Mars colonization, then I suggest that if you find this very sad, then please, the two things that are very good about it. First of all, if there's interesting life on Mars, this will greatly give you lots of interest in sending humans to study Mars from orbit. So, using the Martian moons, Phobos and Deimos, then you can build colonies there, and you can study Mars from orbit, and there will be actually a reason to be there, which will unlock lots of finance to send colonists to Mars, to study scientists, but also scientists just like in Antarctica. When you have scientists, you have all the people that support them as well. So you would get a whole settlement in Mars orbit eventually. And then, as well as that, and then you would study the Mars surface by telerobotics, and I think we should do this anyway. And then, before we land humans there, because we need to have a very thorough understanding of Mars, we could land very quickly with humans in orbit around Mars. What we will be able to discover with sample return is nothing compared with because it's like the entire land area of Earth that we need to explore somehow. And a dozen or more possible habitats. I only mentioned one of them yesterday in my last video. So it's going to be a long process before we find it out. We could greatly accelerate that with lots of people in orbit and with assets on the ground. It would be like exploring the seabed. It's like you're thinking about having a sea colony, but before you have a sea colony, you first explore the seabed and find what's there. Then Mars would be very like that. You're considering having a colony on the land, but before that we have to study and find out what it's like. And you could have very quickly, within less than a decade, you could have sent small rovers, sterilized very carefully throughout Mars, studying it. In my paper I look at ways of sterilizing rovers. There's a way of the ideas for the Venus surface rovers, which could be sterilized. There would be the ways that you can get a rover that works at, I think it's 200 degrees C. Let's look at my paper. Venus. Yes, here we are. 100% sterile rovers. Then there's one with active cooling at 200 to 300 degrees centigrade. That design of rover, it's meant to withstand 300 degrees centigrade. Now we have, you get people saying, oh, with Viking we were able to devise rovers that can withstand very high heat sterilization. We don't have that anymore because our things are so sensitive. That's not true at all. The reason that our spacecraft can't withstand heat sterilization is because we use low-cost components. Naturally, they use the lowest cost component that will do what is needed, and those can't withstand high temperatures. But we have many situations on Earth where we need high temperature components. For instance, in oil wells, aviation, electric cars, very close to the engines of planes. We have many sensors, and they have actually found out that there's enough equipment to design an entire rover that is able to withstand 300 degrees centigrade permanently. So, and they actually say in these studies this could be useful for planetary protection. I think it's a very high priority if there's a possibility of life on Mars, which there does seem to be, then in order to explore it properly, if we keep on sending things like Opportunity and Curiosity and Perseverance to Mars, they're not 100% sterilized. If we start sending these to brine layers on Mars, then eventually they're going to contaminate them with terrestrial life. And Mars life could potentially, and it could be a mix, it could be some life that is very robust and some life that is very fragile, that is very easily destroyed by terrestrial life. For instance, if it had, there's what they call transformable cells, that Carl Russ's idea about life before you got DNA. Before you got Darwinian evolution, you had Lamarckian evolution. We had little what do you call them? These little containers. I'm forgetting the name. Gone from my mind again. But the like lysosomes. That's the wrong word. That's coming up in my mind. These little capsules inside cells. What you call them? Then these with a little bit of genetic material inside them, then could those evolve? Let's look up first, and I'll get you the right word. Yes, small virus-like replicons that populate abiogenic lipid vesicles. That was the word I was thinking about, vesicles. Lipid vesicles. So they could have evolved separately. So you have like tiny vesicles within cells. These could evolve separately, and then the cells would evolve to have to be able to exchange DNA very easily, have totally permeable membranes, where foreign materials just go in and out. Something like that would be very easily destroyed by terrestrial life. It's hard to see that withstanding terrestrial life. So if there's something very vulnerable on Mars like that, then it could just go extinct as a result of including terrestrial life. And it could be a mix of, and even something like this, which is mirror biology, and just a little bit advanced over that, then terrestrial biology, most terrestrial biology would not be able to eat mirror biology. It could be naturally resistant, even though it's quite feeble. And it's not impossible. We have RNA world life on Earth. This was the shadow biosphere hypothesis. It doesn't seem to have been found, but it's biologically possible to have a shadow biosphere, so we could introduce accidentally a shadow biosphere from Mars to Earth. So we have to be very careful that if we introduce a shadow biosphere to Earth accidentally, that it's not good to Earth, that it's not going to be harmful to Earth. As I look in my paper, there are possibilities of ways it could actually be beneficial, and a shadow biosphere on Earth could be beneficial, but it could also be harmful. And we need to know the answer to that before we consider the possibility of introducing, deliberately or accidentally, a shadow biosphere to Earth. A new shadow biosphere doesn't exist here anymore. And so again for the Earth. So then, and if you look at the Mars colonization again, then if you talk about returning a shadow biosphere, but you also, there's certainly a possibility that the surface layers on Mars, they're oxygenated, highly oxygenated surface, temperatures similar to Earth in daytime. there could be biofilms there, then there could be pathogens of biofilms. Those pathogens of biofilms could also be pathogens of human lungs, like Legionnaires' disease. Legionnaires' disease is a pathogen of biofilms. It's also a pathogen of human lungs, an opportunistic pathogen. You also get the possibility of opportunistic fungi, and we have opportunistic fungi, and they cause many deaths actually. And so you have to be very careful with opportunistic fungi for vulnerable people. And so those could come from Mars. And then if our terrestrial biology doesn't recognize it, the antibiotics in our skin, in our epithelium and so on, will not stop it. And below several microns, microplastics can access any human organ. So in the same way, if it's unrecognized by our immune system, then it could access any human organ. So you have to be very careful about that. What would this do? For instance, microplastics, then they develop a halo of blood plasma around them. Now, if you have small amounts, that's no problem. And with microplastics, isn't enough to be a problem. But with large amounts, little bits can stick together. So if you had a situation where much of our biosphere was Martian and was a shadow biosphere, and if the shadow biosphere of our body was basically impermeable to it, like it is to microplastics, then you get the situation where just the blood halos could end up cutting our blood supply and cause heart attacks. That's just one example. And that could happen for Martian colonists. They land on Mars, everything looks fine for a while, but the Martian life starts reproducing inside their habitats, and it gets more and more common, and then their bodies are just permeable to it. And then it could invade their lungs, and they seize their lungs as a nice warm biofilm. So this, I'm just sketching it here. I've got several sections on that in my paper, and I'd like to go through in more detail, and I'll do that in the next video. But what I'm saying here is that it is not pre-decided in advance that it is safe to send humans to Mars. It's not pre-decided in advance that it's safe to return materials from Mars to Earth. Because we've never done anything like this before in entire human history. Nothing like this has happened. And if we look at extraterrestrial species, it's entirely possible that extraterrestrial species have gone extinct doing this. It's not impossible. So we have to be very careful. And if done carefully, it will be of great benefit to us. But if done carelessly, it could be harmful. And so we have to be careful about that. I don't think personally that even if we return neurobiology, I don't think we would go extinct. But we might have to para-terraform Earth. We would have to build habitats covering Earth that have terrestrial biology inside to keep out the extraterrestrial biology outside, if humans are permeable to extraterrestrial biology, and if they're harmful to us, and if they destroy our trees, and they destroy our plants, and they destroy our crops, and destroy our animals, then that's what we would do. And then eventually we'd para-terraform large parts of Earth and parts of the sea. We would preserve all the seeds because we have them in the seed banks, so we'd be able to reconstruct many of the biospheres. We would probably have emergency work to preserve the tropical fruits and other things, and animals and many species and corals and all that sort of thing. We would have it would develop over a number of years, so we'd have time, I think, to preserve our terrestrial biology. But this is not a future that we should aim for. So this is, I think this is, I don't see an actual reason why this can't happen. And I've got some sites that I use. There's again, what's his name? But there's a study of a physicist. I've only got one chance of physicist. Yeah, so this is the idea of diminished of Martian, extraterrestrial Martian life could set up a diminished Gaia on Earth. Not a swan song Gaia. Still be a lot of life here, but much less than we would expect if it wasn't for it. And yes, worst case scenario where there's no defences. I'll talk about that in future, Now, I don't want to go into great detail there, but yes, I'll go into a lot of detail there about that here. You can find that in the paper. So I've got many pages and pages of it. It would take ages to explain all this. So I'm not going to talk about that just now. But that's about the blood plasma that I talked about and how they stick together. Nanoplastics stick together. Anyway, I'm going to talk about it in a separate video. I don't want to get too involved in that right now. And so, what else am I going to talk about here before I end this? So we've got the lab plasma. That's the safest place to return it. Yeah, so I was just saying that Martian colonists, I think, should want to be behind all this. If they're sensible, if they're wise, if they're not just reckless. There's a difference between taking risks and being reckless. And in my view, going to Mars to colonize right now on the basis of what we know now is not just risky, it's reckless. It's not just reckless for the colonists, it's reckless for Earth. And I think it's just not going to be permitted because we have very strong environmental laws to protect Earth. Once this becomes clear, as it will become clear as we study, as we look into the Mars sample return, it becomes clearer and clearer that we can't protect, we can't protect a Mars sample return, Earth from a Mars sample return, if it's something like mirror life, the current technology. And then it becomes very clear that we can't protect Earth from returning Mars colonists. So the answer is to colonize in orbit around Mars and to colonize the Moon. And the Moon is far, far, far easier to go to. I'm a great advocate for lunar colonization. There are many people who have written books on that. The ones who write about Mars colonization for some reason get the press more. But the ones who many people who write about the Moon, and it's a big difference. Ones write about Mars, they have a tiny one chapter. Robert Zubin has one chapter on the commercial use of Mars. And a very vague hand-waving argument saying they'll be able to pay for themselves by export of deuterium. It doesn't work if you look at it close up, because the concentration is just a little bit more than what we have on Earth. And it's just not worth those extra steps to export deuterium from Mars to Earth. And if you look at the only other things they have is export of ideas, but you can do that from the Moon, you can do that from anywhere. You would have export ideas from the seabed, which is a very challenging place to colonize. So export of ideas, because you're living in a difficult terrain, is something you can do from anywhere. And actually the net balance would be import of ideas, because they would have to license all the spacesuits and all the spacecraft and all the things that they are getting from Earth. So I can't see a colony paying for itself by licensing its ideas. But if you go to the Moon, and books about the Moon, they're full of ideas about how to commercialize it. You can sell platinum, you've got tourism, you've got the scientists, of course, you could be scientists going to the Moon, they need to be supported. The science head will be supported on Mars, and eventually you'll get tourism once it's very fast transferred to Mars. But right now you're not going to get tourism. And so you get tourism on the Moon, and that'll be a very big finance. Once we got to the point where I think we're going to be able to fly 1 billion people into space. Eventually, once we've got, I'm not sure that rockets are going to be a thing. Maybe it could be rockets, maybe very, very big rockets. Maybe it's the Skylon and that Europe is betting on, of sending planes to orbit. One way or another, we're likely to have billions of people flying into space, just like we have billions of people flying around Earth in the near future. So where else do they want to go apart from Earth? The Moon. So I think there's going to be a big tourism industry in the Moon in 10, 20 years from now. And then there's all the science interest. There's the ice on the Moon that could be exported to Earth. There's platinum is possibly there. There's quite good evidence that it could be from the core of the huge impactor Aitken crater. It's splashed out onto its rim. There's magnetic anomalies that may be signatures of high concentrations of platinum. There are lots of commercial possibilities for the Moon, and there's also the possibility of the Hoyt transfer, which lets you transfer material from the Moon by a rotating tether and then to another tether above Earth, with actually gaining energy through the gravity well from the Moon to Earth. It's very ingenious. You can do that from Mars too, in a way. You can have rotating tethers involving Phobos and Deimos. You can have an elevator. You could try rotating tethers, but it's far, far more difficult than from Moon to Earth to export without having to use lots of rocket fuel. So, but eventually, in this worst case for Mars colonization, actually a very good case for astrobiological interest, then if you can't send, if we discover that what's on Mars is just so hazardous to Earth's environment that you can never send humans from Mars back to Earth, then we can still export from Mars if we find something there that is worth exporting. We do it by telerobotics. And the telerobotics about Mars is going to be very interesting. If you look at the telerobotic orbit, the arrow orbit that it does, twice a day. It'll come low in above Mars, then skim over the poles and go out again. Where we are, it becomes like a small planet in the distance, and you come in again, and you see it from orbit again, and you're very close, and then you have several hours of telepresence, and then during telepresence, you would be like a gamer into playing one of these 3D games with Kinect and with a omnidirectional platform to walk on. And so you could walk around on the Mars surface with miniature or huge or strong or whatever avatars on the surface, and then you would see it actually more clearly than you could with your human eyes on the surface, because your vision could be adapted to see whatever you want to be able to see most clearly. And you'll be able to fly, you'll be able to go into tiny, tiny caves and so on using avatars. And we're already doing this sort of thing. We already have the possibility of remote surgery by avatar. It's actually been done, experimentally it's been done. Surgery in France by the surgeon, I think it was in the United States or something. And then this is only just beginning in space, but I think we're going to have telepresence, this exploration of Mars from orbit, and Mars is going to be far more interesting for telepresence exploration if we preserve the native biology, whatever it is. If there's mirror life there, if there's early life, and if it doesn't go extinct, and we don't have it contaminated with terrestrial life, then I think we're going to have a much more vibrant Martian community in orbit than if we just land 100,000 people or whatever it is there. They try and make a going. Mars is not good for agriculture. You can't grow anything on the soil of Mars. At night, it goes to dry ice temperatures. Dry ice actually forms on the surface of Mars, even in equatorial regions some parts of the year. And then the atmosphere is just too dry you're never going to get, most plants are just not going to be able to survive. Just some lichens that are able to somehow use the moisture in the air and these salty brines. It's not much good for agriculture. So literally the only way you can do agriculture on Mars is in greenhouses. But if you take a standard greenhouse and put it on Mars, it'll instantly explode because of the pressure inside. We don't realise how high the pressure is inside a greenhouse, but it's because it's counteracted by the pressure on the outside, but it's tons per square metre. 10 tons per square metre if it's at full atmospheric pressure. If it was a fifth atmospheric pressure, it would be 2 tons per square metre. So this requires very robust construction of your greenhouses, and your houses have to have like a metre of regolith. There's just no way that it makes sense to do agriculture on Mars as a way to just go out and you want to do like frontier agriculture, your log cabin thing, your frontiersman. It doesn't work as agriculture. You have to have another reason to go there. The interesting astrobiology is a good reason to be on Mars, but it would be from orbit. I think that the most interesting astrobiology discoveries will come if we never land on Mars. If there is interesting biology, even if it's related to terrestrial life, we want to know how it's related, what the differences are. And whether you eventually decide to land humans on the surface and start building your colonies, but we can right away, there's no problem at all, very easy access. We haven't been back to the moon. I think moon colonies are the way to go first. And the lava tube caves, which are absolutely vast, they may be. We know they look very long. They are certainly tens of meters in diameter. They may be as much as kilometers in diameter in the low lunar gravity. There's the ice at the poles. There's the very stable temperature at the poles. There's the nearly 24/7, nearly year-round solar power at the poles. So for all these reasons, I think the Moon is a far more attractive place to attempt settlement than Mars. And then once we've done that, then we can decide where we go next. There are many other places like Callisto in the Jupiter system, and there's Titan in the Saturnian system, if we can do that in a planetary protection friendly way, which is possible. So I do talk about that briefly in my paper as well. So, to go about an hour, and I'll sort of gradually wrap up. Is there anything that I haven't said yet? So basically, I'm trying to bring Mars. I was a Mars colonization. I still am a colonization enthusiast. And I used to think, oh, great, we should go and colonize Mars. I just didn't even, unreflecting, 10, 15 years, 15 or 20 years ago, if you'd asked me, I'd say I wouldn't see any problems with it. But then I came across, and I also at the same time knew that we had to sterilize rovers to go to Mars. And I just never made the connection. And it's amazing how our minds can be compartmentalized for decades. We just don't make that connection. Way back, I mean, and the way back 1960s with lunar landing, you knew from then that you need to protect planetary protection. So I watched the lunar landing and saw all that. And yet, all the decades from then, it's only about 10, 15 years ago, that I started to realise that humans on Mars would you need to look at planetary protection for that, just as you do for rovers. And so that is what eventually led me to this journey that led to this. And I will just spend to let me writing this paper, and I'm just going to show you my Touch Mars article, which is, so then I started doing blog posts, and I wrote this booklet, which you might enjoy reading, especially if you're less specialist. Paper is not so technical. I'll wait for it to come up. So this is where these ideas came from. I started off by I think the main place. So my research interest was in the logical foundations of mathematics and set theory. So I trained in logical reasoning and different forms of mathematical reasoning, including intuitionistic logic and classical logic, and I devised a new logic in between the two. And I hope someday to publish it. But there were problems with I did this at Oxford, and my supervisor, Bob Gandhi, he sort of supported me all the way through. But then when it came to the examination, then sadly the reviewers, the examiners, they rejected it, and they did that twice. And then my supervisor said, okay, what you should do now is to do a doctoral by research, where you just publish the papers and don't try and get it examined by anyone. Just get it, send them for peer review, and then once you've published enough papers by peer review, then you can get your doctorate just by there's a DSc instead of a PhD. But sadly I got discouraged and I just never did that. I still have these papers that I want to write and to submit for my DSc sometime. But that's my background, is in logic and set theory. And so the main way, a long-term interest in science, long-term interest in astrobiology, the main way I come with this an overview and I've got very clear understanding of how logical processes work. And so the logical analysis of all the different situations that have been, and I can read papers very easily and get the structure of the papers and understand what the paper is about in many different disciplines. Again, I think that's because of my logical background, as well as very long-term scientific interests. And so that's why I think it's just like I had this compartmentalizing. We've got the compartmentalizing in planetary protection. The people researching one thing, people researching another thing. People researching into making these sample retrieval facilities are not the astrobiologists, they're engineers, and then they get a typo difference. They think they've got to design it down to 0.1 microns when it's 0.01/0.05. So you get these things. And so I think the main way that I'm helping is by having an overview of the whole process. I don't have detailed understanding of any particular area, but I have this overview, and that I think is the main thing that I can contribute. But this, because my paper, and then as I said in the previous one, the natural thing to do would be to just do one short paper, another short paper, another short paper, bit, then build up until you've got it. But this is something that is due to happen very soon, and I just don't see there's time to do that. So I'm getting together this rather long, involved paper with multidisciplinary and really rather it can be very difficult to peer review, I would imagine. But I'd like to get some discussion about it, even if my paper is never accepted for publication, or it's just delayed and delayed and delayed for years, then still we need to talk about this. If I'm not going to be doing papers, other people need to publish the papers. But I'm going to try and publish it to peer review, and I'm very interested in your comments on it, and just any ideas you have about it, anything that I'm missing, and anything that needs to be added, anything, any mistakes, please say. And I very much appreciate if you've had the patience to listen to these 2 talks that I've just given, and I hope you enjoyed them. I hope they were interesting, and I very much appreciate anything that you have to say about what I've said. And so that's it. I'll upload this video as well, and I'll upload both videos. I've uploaded this one to the wrong account. Oh dear! I'll have to re-upload it to the right account. Anyway, so I'll stop now.